So I am starting the class 1 part 2, and in this class part we are going to study the search of genetic material and the rest of the portions, including, let us check what is there after this, the RNA world. Okay, so these are the two parts we are doing, the search of genetic material. Okay, even though the discovery, see, I have told that nuclein, it was discovered by Meister, and it took very long to understand the structure of DNA. I have already discussed this. Now, Mendel, almost at the same time, had given the principles of inheritance. Okay, principles of inheritance, there were, you know that Mendel has proposed Mendel's laws. Okay, there are three such laws: law of dominance, law of, so all these, there are three laws which Mendel has given. Okay, so DNA act as genetic material, it took very long to be discovered. Now, by 1926, obviously years are not important. Okay, this is not UPSC, it is NEET. So for NEET, you don't need years. Okay, so mechanism for, so the what will be the basis for the mechanism of the genetic inheritance? So by 1926, it has reached to the molecular level. Previously, Mendel or Sutton or Morgan and numerous other scientists have narrowed the search to chromosomes located to nucleus, but the question, what molecule was the genetic material, had not been answered. So these three names, Mendel, Sutton, Morgan, they narrowed the genetic material search to chromosome. But what chromosome was, what was the molecule that is the genetic material, that was not answered. Now comes the transforming principle, very important. Transforming principle is very, very important. So Griffith, we call it Griffith's transforming principle. Okay, we also call it Griffith's transforming principle. It is a series of experiments. So what was used? For this, Streptococcus pneumonia. It is wrong. P should be small. Okay, so all these writings very important. It is the bacteria which is responsible for pneumonia. So it was used. Okay, so when Streptococcus pneumonia or pneumonococcus, they were grown on a culture plate with some smooth shining colonies. Okay, so there are two types. One will produce smooth shining colonies S, and others will produce rough colonies R, because S strain bacteria have So what is this smooth shiny thing? What is this? That is actually a mucus coat, and R doesn't have. Okay, this A strain, there is two type of strain, A strain and R strain. So A strain is smooth shiny and R strain is rough. Why it is smooth shiny? Because it is having a mucus coat. This mucus coat makes it virulent. Virulent means that means if this A strain will be injected to mice will die. Okay, so A strain inject to mice die. This is called virulent. Okay, but R strain is non-virulent. It is injected to mice is living. Okay, A strain, when heat killed, it will be injected to mice, so A strain is there. Now when I am heat killing it, that means, and that time it is becoming what? Non-virulent. How I can say it is becoming non-virulent? This is R strain is also non-virulent. See, mice are living, that means it is non-virulent. So A strain heat kill, which is non-virulent, and R strain leave. R strain leaving is also non-virulent. I am injecting to mice. So what? Both are non-virulent. So mice should live. But here mice is dying. Why? So this was the observation. So what did he conclude? That something has transformed. The R strain bacteria has been transformed somehow by heat kill S strain bacteria. R strain bacteria, they were non-virulent. Okay. S strain heat killed. They were non-virulent. A strain bacteria has somehow transformed R strain bacteria to form this smooth polynucleoside coat, or the mucus coat which I was talking of. This mucus coat, mucus polysaccharide coat. Smooth mucus polysaccharide coat. And by that, S strain has become virulent. And it is when both are injected to mice, mice is dying. However, the biochemical nature of genetic material, it was still a mystery. Right? Now, see this thing very carefully. The biochemical characterization of transforming principle. Now see, transforming principle was proposed by Griffith, but biochemical characteristics, there is nothing to do with Griffith. Now another person is coming into action. Other three scientists, Avery, MacLeod and McCarthy. The genetic material was thought to be, they worked on Griffith's experiment. Before their work, everyone was considering protein as the genetic material. But these three scientists, Avery, MacLeod and McCarthy, after their work, biochemical nature of transforming, they actually observed the biochemical nature of the Griffith transforming principle. Okay, so they purified the biochemicals. What? Protein, DNA, RNA. So protein was there, DNA was there, they separated this, isolated this, RNA was there. Okay, from heat kill H cells, and they were checking that which will transform R cells into S cells. I told you that R cells were transformed to S cells and then they were becoming virulent and then they were killing. I told you this. So which can do this? Protein, DNA or RNA? So they discovered that, so what can I do? This mixture is there. So I will once check with, I will give protease. It will digest the protein and see that this phenomenon is occurring or not. So when protease was given, nothing happened. When RNase was given, what should have happened? When RNase is given, see, when RNase was given, what should happen? This transformation should have stopped, but no, the transformation, nothing happened. But when DNase was given, then DNase will digest the DNA. Then actually this process was stopped. Now S strain heat kill was not able to transform R strain. That means DNA caused the transformation. So DNA is the hereditary material. But by all these things, all the biologists, they were not convinced. So the genetic material is DNA. Who? Now Hershey Chase experiment. Finally, Martha Chase and Alfred Hershey. So Hershey, you eat Hershey chocolates, right? But no, it is different. So Hershey Chase experiment, finally they actually proved that the genetic material is DNA. How? We will discuss. Here is the experiment. See, this is the experiment. Oh, sorry. This is the experiment. If you see this, you will understand everything. And this is the whole experiment. See, I am teaching you the experiment. What is happening? So see, these are the two bacteriophages. This is one bacteriophage. This is another bacteriophage. Right? Now, this bacteriophage, this bacteriophage is grown in a culture medium. Suppose this is a culture medium, okay, and this is the bacteriophage, okay. Like this, there are various bacteriophages grown. One to cannot be isolated. So this bacteriophage, it is grown in 35S radioactive sulfur, okay. So this sulfur, it will get incorporated to the protein part, protein capsule, and in another culture medium of 32 in radioactive phosphorus, another set of bacteriophage was developed. In this, the DNA is radioactive. So in this bacteriophage, DNA is radioactive. In this bacteriophage, protein is radioactive. Now both were allowed to infect a bacterial cell. Bacteriophage is a virus which can attack. What is the meaning of bacteriophage? A virus which can attack bacteria. Okay. They were allowed to infect a bacteria. See. Now they were separated. Okay. Now here there is a little bit of, this diagram has a little bit of problem. See, this should be red in color. This is one of the printing mistakes in, see. So here is the radioactive. Okay, no, sorry. It was in the old NCERT, this problem was there. New NCERT don't have this problem. So see here, the protein capsule, this is radioactive, right? So the DNA is not radioactive, right? So DNA is not radioactive, right? So no radioactive detected in cells because DNA was not radioactive. But this is called supernatant. Radioactivity in the supernatant, obviously, because protein is in the bacteriophage. Here you see the radioactive DNA getting incorporated. So radioactivity will be detected in cells. Okay. That means DNA has been transferred, right? Here you can see they were seeing what? Now let me tell you. They were seeing there was a bacteriophage. One bacteriophage was there which was having radioactive DNA. Another bacteriophage was there. Another bacteriophage was there which is having radioactive protein. Okay. This was allowed to attack one cell. This was allowed to attack one cell. Now the one which has DNA, this bacteria turned radioactive. And this bacteria, nothing happened. That means the bacteria which was infected by the bacteriophage, which I am telling you this in Bengali for better understanding, যে ব্যাকটেরিওফাজে রেডিওঅ্যাক্টিভ ডিএনএ ছিল সে হচ্ছে ব্যাকটেরিয়াটাকে রেডিওঅ্যাক্টিভ করে দিল, কিন্তু যে ব্যাকটেরিওফাজের প্রোটিনটা রেডিওঅ্যাক্টিভ ছিল সেই ব্যাকটেরিওফাজটা যে ব্যাকটেরিয়াটা থেকে অ্যাটাক করলো সেই ব্যাকটেরিয়াটা কিন্তু রেডিওঅ্যাক্টিভ হলো না। তার মানে প্রোটিন, এরও তো ডিএনএ ছিল, তার মানে প্রোটিন ট্রান্সফার হয় না, ডিএনএ ট্রান্সফার হয়। তাই ডিএনএ ট্রান্সফার হয়ে গিয়ে পুরো ব্যাকটেরিয়াটাকে রেডিওঅ্যাক্টিভ করে দিয়েছে। এইটা হলো সিম্পল সেন্ট্রিফিউগেশন। তার 3টে স্টেপ আছে Hershey-Chase experiment. There are 3 steps in Hershey-Chase experiment: infection, blending and centrifugation. Now we will discuss the properties of genetic material DNA versus RNA. Okay. So properties of genetic material DNA versus RNA. So now it is resolved that DNA is the genetic material, not the protein. But subsequently it became clear that RNA is also genetic material for some viruses like tobacco mosaic virus and QB bacteriophage virus. So answer to such questions such as why DNA is predominant genetic material, whereas RNA performs dynamic function of messenger and adapter. See, RNA has various functions, I have told. Main function is messenger, but along with this it has functions like adapter molecule, it has functions like structural molecule, and also another function, the name of the function is what? Structural adapter and catalytic molecule. Okay, so there are so many functions of RNA. Okay, so differences between chemical structures of two nucleic acid molecules, that means difference between DNA and RNA, which I have already discussed. So a what are the characteristics of a genetic material? This is also very, very important. What are the characteristics of genetic material? Number one, it should be generate its replica. That means it should undergo replication. Okay, now it should be, second point is it is chemically and structurally it should be stable. Okay, and the third point is that it should provide scope for slow changes. That means it can mutate. So gene mutation is taking place, right? So see, this mutation is very important. If the genetic material will not mutate, then evolution is not taking place. So evolution should take place. And the last is it should be able to express itself in the Mendelian characters. Okay, now if one examines each requirement, now we will examine each requirement one versus one for DNA and RNA, and they are to direct their duplication. Other molecules such as proteins fulfill this criteria. So first point is applicable for both DNA and RNA. A replication DNA also can do, RNA also can do, but protein, no, protein cannot do it. Okay, now the genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. So stability is one of the properties of genetic material that was evident in Griffith's transforming principle. In the Griffith's transforming principle, you see heat. See, stability is very important, that in the various stages of life cycle or during age, if there is a change in physiology, it should not change. And so from Griffith's transforming principle, we are saying that heat was able to destroy other things but not the DNA. So DNA is a very stable material. Okay, so this explained in the light of DNA that first of all, the two strands of DNA are complementary. If separated by heating, they will come together. So the two strands of DNA can be separated by heating, but the two strands of DNA, they are joined by hydrogen bond, they will again come back. Okay, and the two, I told you that in DNA, in RNA there is a 2' at OH, at a 2' OH is present. 2' OH means in the 2' carbon OH is present, and this is making RNA a reactive group. So RNA is liable and easily degradable. RNA is also known to be catalytic, and if it is catalytic means it is reactive. Okay, and DNA is therefore more stable and less reactive in nature, right? So it is very much suitable for the genetic material. In fact, presence of thymine, again more stability from where we are getting. So one complementary strand we are getting stability. Second, the 2' OH at the RNA, it is making RNA unstable and reactive. Third, presence of thymine in place of uracil also gives additional, see, DNA becoming additionally stable because of thymine, and uracil in RNA is making it more reactive. Okay, or less stable, both RNA and DNA can mutate. So third point is only second point is only applicable to DNA, not RNA. Okay, third point, mutation. Mutation, both RNA and DNA can mutate. Okay, and see, RNA, consequently virus, in fact RNA is being unstable, mutated faster. RNA, because as it is unstable, it is reactive in nature. Unstable means it is reactive. It is reactive in nature, so it can mutate at a faster rate. Viruses having RNA genome have shorter lifespan, mutate and evolve faster. Okay, so for viruses the genetic material is RNA. So viruses, they have very short lifespan and they mutate faster. You can see for the coronavirus or the COVID, this virus, there are so many mutations. Each and every form we are getting, one is Omicron, one is beta, one is alpha, one is delta. Okay, so RNA can directly code for synthesis of protein, hence can easily express characters. DNA, however, is dependent on RNA for synthesis of protein. So okay, so RNA, it can form protein directly, but DNA is dependent on RNA for protein synthesis. Very, very important. So the protein synthesizing machinery has evolved around RNA. Therefore, indicate both RNA and DNA can function as genetic material, but DNA, being more stable, is preferred for storage of genetic information, for transmission of genetic information. RNA, so DNA has stability. This is the main point why DNA is genetic material. DNA is known for stability, so it can store genetic information. And RNA, what is there for RNA? RNA can transmit genetic information. So both has different functions. Okay, now we are going to study the third part, that is the RNA world. From foregoing discussion, an immediate question comes to evident, which is the first genetic material. It shall be discussed in detail in the chapter on chemical evolution, that is, but briefly we shall highlight some of the facts and points. See, RNA was the first genetic material, very important. Okay, there is now enough evidence to suggest essential life processes such as metabolism, translation, splicing evolved around RNA. So for RNA, you can see these phenomena. You can see metabolism, you can see translation, you can see splicing. Okay, RNA used to act as a genetic material as well as a catalyst. So RNA is both genetic material and catalyst. Okay, there are some important biochemical that are catalyzed by RNA catalyst, but not by protein enzyme. So there are certain biochemical reaction which are being catalyzed by RNA catalyst, not the protein, not the enzymes. Okay, RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that was more stable. So now we are talking about evolution. First, there was RNA, and that was the genetic material. In that time, organisms, see, from one organism, another organism is coming. Like this evolution is taking place. Year after year, the evolution is taking place. So in Earth, the organisms which were there, the viruses or whatever the information, the prokaryotes, whatever was there, that time the genetic material was RNA. Now, these RNA, they were acting as both catalyst and they were acting as the genetic material. Now, RNA, why they are acting as catalyst? There are certain biochemical reaction which was carried out by RNA catalyst. They can undergo metabolism, translation and splicing. That is also clear. Now, DNA has evolved now, but RNA, there was one problem, that is the stability is the issue. It is very reactive. So from RNA came DNA. Now this DNA by some structural changes. What are the structural changes? First, thymine in place of uracil. Okay. Second, what is another structural change? The two in the RNA there was at 2' end there was OH, so this was gone. In case of DNA, this was gone. Okay, so these are the major differences, okay, which were responsible or DNA, and I have to discuss you what are the differences between DNA and RNA. So that modifications took place, and DNA and being double stranded complementary further resist the changes by the process of repair. Okay, so now we will study replication. So this is the diagram of replication, but we will study replication and other part of the chapter in the second class 2. Okay, and class 2 how many parts will be there, that I will say to you at the beginning of class 2. So study up to this much, and whatever the questions which have been told you to do as assignment, please do it. The final, after the completion of the chapter, the final assignment of solving MCQs will be given and has to be submitted within 2 days, that is later on. So up to this much, thank you. This is all about class 1. We will resume the class 2 at the scheduled time.